Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with tardigrades, the microscopic travelers who survive the vacuum. Nhalman, the Nastrova, Heike Veals, they breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. These manate organisms, often referred to as water bears or moss piglets, belong to a phylum known as Tardigrade, with a length typically ranging from 0.5 to 1.5 millimeters. They inhabit diverse environments, from the deepest ocean trenches to the highest mountain ranges. Their resilience allows them to endure extreme conditions, including temperatures from absolute zero to over 150 degrees Celsius. Tardigrades possess a unique body structure, characterized by a segmented body and 4 pairs of stubby limbs, each equipped with claws. This morphology aids in their ability to navigate through various substrates, whether it be moist soil, lichen, or leaf litter. These organisms are equipped with a chitinous cuticle that protects them from desiccation and environmental stresses. In addition to their anatomical features, tardigrades exhibit a remarkable ability to enter a cryptobiotic state, effectively suspending their metabolism. In this state, they can survive without water for years, only to rehydrate and reactivate their biological processes when conditions improve. It is this extraordinary adaptation that enables them to survive in the vacuum of space and extreme radiation levels, drawing significant interest from scientists studying the limits of life on Earth and beyond. Their presence across various ecosystems underscores their integral role in the microbial landscape. The initial discovery of tardigrades dates back to 1873, when the German zoologist Johann August Ephraim Goetze first described these microscopic organisms. He referred to them as little water bears, a nod to their unique appearance. Early research focused on their habitat, revealing their affinity for mosses and lichens, environments teeming with moisture. Observations demonstrated their remarkable ability to withstand extreme conditions, igniting curiosity among scientists. Subsequent studies in the late 19th and early 20th centuries expanded understanding of their biology. Researchers began to employ microscopy techniques, providing clearer images of their anatomical structures. It became evident that these creatures possess a unique ability to enter a state of cryptobiosis, allowing them to suspend their metabolic processes under desiccation or extreme temperatures. This phenomenon prompted further investigation into their resilience and adaptability. In the decades following, advancements in technology led to more comprehensive examinations of their cellular mechanisms. Tardigrades became subjects of interdisciplinary studies, attracting the interest of ecologists, biologists, and astrobiologists alike. The discovery of their presence in some of the most extreme environments on Earth, such as deep-sea trenches and high-altitude regions, established them as key organisms in the study of extremophiles. This growing body of knowledge continues to shape current research, revealing insights into life's potential beyond our planet. Taxonomy reveals the intricate classification of tardigrades, a group that occupies a unique position within the broader tree of life. They belong to the phylum Tardigrade, which is distinct from other invertebrate groups, categorized into several classes. The most prominent classes include Eutardigrade, Heterotardigrade, and Mesotardigrade. Eutardigrade encompasses the well-known water bears, which are frequently studied for their remarkable resilience. This region of life is further divided into over 1,000 identified species, each displaying a variety of morphological characteristics that contribute to their survival strategies. They possess a segmented body, typically comprising 4 pairs of stubby legs, which are equipped with claws that allow them to navigate through diverse environments. Their unique anatomy includes a cuticle that can withstand extreme conditions, ranging from intense radiation to extreme temperatures. This resilience is partly due to their exceptional ability to enter a cryptobiotic state, effectively suspending their metabolic processes when faced with desiccation or high radiation levels. The classification of tardigrades extends to their evolutionary relationships, revealing a lineage that has endured for more than 500 million years. Genetic studies continue to uncover the complexities of their evolution, providing deeper insights into how such organisms thrive under conditions that would be hostile to most life forms. Morphology reveals a fascinating array of physical characteristics that contribute to the resilience of tardigrades. These minute creatures typically measure between 100 and 500 micrometers in length, with a body composed of a segmented structure. The cuticle, a protective outer layer, is often adorned with intricate patterns that vary across species, hinting at their evolutionary adaptability. Until short limbs extend from their bodies, each ending in tiny claws or suction pads, enabling a remarkable range of movement. This arrangement enhances their ability to navigate through diverse environments, from moss to the deep sea. The mouth, equipped with specialized stelates, allows for the consumption of plant cells, small invertebrates, and microorganisms, showcasing their versatility in feeding habits. Our unique feature lies in their capability to enter a cryptobiotic state. When faced with extreme conditions such as desiccation or radiation, the body can retract and curl into a ball, significantly reducing metabolic activity. In this state, they lose almost all water content, allowing them to endure the vacuum of space and the pressures of the deep ocean. The internal structure contains a variety of specialized cells, including those that facilitate the repair of DNA damage. This resilience is particularly notable under stress, underscoring the incredible adaptability of these organisms in the vast tapestry of life on Earth. On Earth. These sizes and shapes vary considerably among tardigrades, reflecting their adaptability to diverse environments. Typically measuring between 100 micrometers and 1 millimeter in length, these minute creatures exhibit a remarkable range of forms. Some display a plump, barrel-like appearance, while others are elongated or flattened. The variability in morphology is linked to their habitats, with species evolving distinct physical characteristics to thrive in specific conditions. In aquatic environments, certain tardigrades possess streamlined bodies, enabling efficient movement through water. Conversely, those inhabiting mosses and lichens often exhibit a stouter form, allowing them to better navigate the dense, fibrous structures of their surroundings. The development of specialized appendages, such as claws or sucking tubes, adds further diversity to their forms, aiding in locomotion and feeding strategies. Mucoloration can also differ widely, with some species exhibiting transparent bodies that allow for the observation of internal structures. Others display vibrant hues, a result of pigmentation that may play a role in protection from ultraviolet radiation or predation. The unique adaptations in size and shape emphasize the evolutionary success of tardigrades, enabling them to inhabit the most extreme of environments, from the depths of the ocean to the heights of mountains and even the vacuum of space. Their presence serves as a testament to nature's ingenuity in the face of adversity. These remarkable organisms inhabit a diverse range of environments, showcasing their adaptability to extreme conditions found in the most unexpected locales. They thrive in moist environments such as mosses, lichens, leaf litter, and soil. The microhabitats they occupy are often rich in organic material, providing essential nutrients for survival. These regions, typically overlooked by larger organisms, create a unique ecosystem where tardigrades can flourish. In addition to terrestrial habitats, they are also present in aquatic environments. Freshwater bodies, including ponds and streams, harbor these tiny travelers among sediment and algae. This aquatic life allows them to absorb water and nutrients essential for their metabolic processes. Saline ecosystems, such as tidal pools and marine sediments, also host these resilient creatures, demonstrating their remarkable ability to withstand high salinity levels. Extreme environments offer further insight into their resilience. Tardigrades have been discovered in the icy depths of Antarctica, illustrating their capability to endure freezing temperatures. Their presence in hot springs speaks to their tolerance for high temperatures as well. These findings indicate a remarkable breadth of habitat, revealing that they can survive in conditions previously considered inhospitable. The adaptability of tardigrades continues to intrigue scientists, prompting further exploration into the mechanisms behind their survival across various ecosystems. Feeding habits reveal a remarkable aspect of tardigrades. These microscopic organisms primarily consume plant cells, algae, and small invertebrates. Utilizing a specialized mouthpart known as a stylet, they pierce the cell walls of their food sources, allowing them to extract vital nutrients. This feeding mechanism enables them to thrive in diverse environments, from the lush mosses of temperate forests to the arid conditions of deserts. In foggy settings, they often feed on algae and bacteria suspended in the water. The abundance of these microorganisms provides a rich diet, allowing tardigrades to flourish in even the most challenging habitats. Their role in the ecosystem is significant. By grazing on these microorganisms, they help regulate populations, contributing to the overall health of their environment. The digestive system of tardigrades supports their efficient feeding. After the style penetrates a food source, enzymes break down the ingested material, facilitating nutrient absorption. This process underscores their adaptability, enabling them to extract energy and sustain themselves in various conditions, including extended periods of desiccation. Research into their feeding habits continues to provide insights into their ecological roles and interactions with other organisms. Understanding these processes can illuminate the delicate balance within ecosystems, showcasing the resilience and versatility of life, even at the microscopic level. Reproductive strategies in tardigrades are as diverse as the environments they inhabit. These microscopic entities exhibit a range of modes, including both sexual and asexual reproduction. The sexual process typically involves the release of sperm into the environment, where it can fertilize eggs laid by females. In many species, this occurs via direct contact, though some rely on external fertilization methods. This method allows for genetic diversity, which is essential in adapting to varying ecological conditions. Asexual reproduction, on the other hand, often occurs through parthenogenesis, where females produce viable eggs without fertilization. This reproductive strategy can lead to rapid population increases in stable environments, facilitating the colonization of new territories. The ability to alternate between these reproductive modes enables tardigrades to thrive in myriad habitats, from the depths of the ocean to the harshest terrestrial settings. Environmental factors play a crucial role in determining the reproductive strategy employed. In times of stress, such as extreme temperatures or resource scarcity, many tardigrades revert to asexual reproduction, ensuring the survival of their lineage. This adaptability reflects their overall resilience, affirming the survival of their lineage. This adaptability reflects their overall resilience, allowing them to endure conditions that would be lethal for most other forms of life. The reproductive mechanisms of tardigrades underscore the remarkable capacity for survival inherent in even the smallest organisms. The life cycle of tardigrades unfolds through several distinct stages, affording to Euler's the remarkable capacity for survival inherent in even the smallest organisms. The life cycle of tardigrades unfolds through several distinct stages, each contributing to their resilience. Initially, eggs are laid in a protective casing, often affixed to a substrate. This encapsulation safeguards the developing embryos from external stressors, enabling them to endure extreme conditions during early development. Upon hatching, the larvae emerge in a juvenile form, resembling miniature adults. This stage, known as the instar phase, showcases the organism's adaptability as it begins to feed. Tardigrades consume organic matter, including algae and moss, facilitating growth and development. This feeding behavior is crucial, ensuring that they accumulate the necessary energy reserves for the subsequent phases of life. As growth continues, they undergo a series of molts, shedding their exoskeleton to accommodate their increasing size. This molting process is vital, allowing for not just physical expansion, but also the renewal of tissues and the maintenance of health. The duration of each stage can vary, influenced by environmental factors such as temperature and humidity. Eventually, they reach maturity, capable of reproduction and continuing the cycle. This progression showcases the adaptability of tardigrades, enabling them to thrive in diverse ecosystems, from the deep sea to the highest mountain ranges, emphasizing their role as resilient survivors in the natural world. Cryptobiosis serves as the remarkable mechanism through which tardigrades endure extreme conditions. This state allows them to enter a form of suspended animation, effectively halting metabolic processes. Within this dormant state, they can withstand temperatures ranging from nearly absolute zero to well above 100 degrees Celsius. This remarkable resilience extends to pressures far beyond those found in the deepest oceanic trenches and to the severe vacuum of outer space. Upon encountering desiccation, a fauna 16 or sparsix asord de verlevely soars the strain a tiwi a sophos or pancounds. Upon encountering desiccation, these organisms lose almost all their body water, entering a cryptobiotic state known as anhydrobiosis. In this form, tardigrades retract their limbs and curl into a ball, significantly reducing their surface area. This minimizes loss of remaining moisture and protects their cellular structures. With protective proteins and sugars, they stabilize cellular components, safeguarding them from damage. Upon rehydration, metabolism resumes, allowing them to reactivate and reproduce. This ability enables them to survive conditions that would be lethal to most forms of life. In laboratory studies, tardigrades have demonstrated survival after exposure to radiation levels that would eradicate other organisms. The intricate biochemical pathways involved in cryptobiosis reveal the complexity of these microscopic travelers, who possess an unparalleled capacity to endure the harshest environments, contributing to ongoing research into life's resilience on Earth and potentially beyond. Anhydrobiosis represents a remarkable adaptation, enabling survival during extreme desiccation. When faced with the loss of water, these microscopic entities enter a state of suspended animation. Metabolic processes are nearly halted, allowing them to withstand conditions that would be lethal to most other forms of life. This region, where moisture becomes scarce, becomes a temporary refuge for them. Upon encountering anhydrobiosis, they produce specialized proteins and sugars that stabilize cellular structures, cellular structures, effectively protecting their vital components from damage. The presence of trehalose, for instance, plays a crucial role in this process. This sugar binds to proteins, preventing them from denaturing and ensuring that cellular functions can resume once moisture is reintroduced. In this dormant state, they can endure extreme temperatures, radiation, and even the vacuum of space. Some studies suggest that these microscopic travelers could survive in the harsh conditions found on other planetary bodies, such as Mars or the icy moons of Jupiter and Saturn. Their resilience raises fascinating questions about the potential for life beyond Earth. Research continues to investigate how the mechanisms of anhydrobiosis might inform biotechnological advancements, such as preservation techniques for sensitive biological materials. In understanding these processes, insights into the limits of life itself are revealed, extending our knowledge of resilience in the universe. Cryobiosis demonstrates a remarkable adaptation to extreme conditions, allowing tardigrades to survive freezing temperatures that would be lethal for most organisms. This state of suspended animation occurs when temperatures drop, causing metabolic processes to slow dramatically, as ice crystals form in their surroundings. These microscopic beings enter a cryptobiotic state, effectively halting their biological functions. During cryobiosis, the cells lose water, and protective proteins prevent damage from ice formation. They can endure temperatures as low as -250 degrees Celsius, remaining viable for decades, if not longer. In this state, they become nearly indestructible, able to withstand not only the cold but also desiccation and high radiation levels. Research has shown that tardigrades can rehydrate and reactivate their biological processes once conditions become favorable again. This resiliency leads to questions about the potential for life beyond Earth, particularly on icy moons such as Europa and Enceladus, where subsurface oceans exist beneath thick ice crusts. The survival strategies of these organisms suggest that life may persist in environments previously thought inhospitable. Their ability to endure the harshest of climates speaks to the fundamental principles of biology and the myriad ways in which life can adapt, challenging the boundaries of our understanding of living systems across the cosmos. Osmobiosis represents one of the remarkable adaptations observed in tardigrades, particularly in relation to their tolerance to high salinity environments. This phenomenon showcases their ability to thrive in conditions where many organisms would falter. Elevated salinity levels create osmotic stress that typically dehydrates cells, yet tardigrades possess specialized mechanisms that allow them to regulate internal osmotic pressure. These microscopic travelers utilize a process known as anhydrobiosis, allowing them to enter a desiccated state under extreme salinity. In this state, metabolic processes slow dramatically, enabling them to withstand the dehydrating effects of their environment. This survival strategy is not merely a passive response. It involves the synthesis of protective proteins and the accumulation of compatible solutes that stabilize cellular structures. Investigations into their genetic makeup reveal the presence of unique genes that facilitate osmoregulation. allowing them to maintain cellular integrity despite fluctuations in external salinity. Such adaptations illuminate the potential for life in brine pools and hypersaline lakes, environments once deemed too extreme for survival. In essence, osmobiosis exemplifies the extraordinary resilience of tardigrades. Their capacity to endure high salinity expands the horizons of astrobiology, suggesting that life may flourish in previously unimagined locales, far beyond the confines of Earth. Radiation resistance stands as another remarkable feature of tardigrades. These microscopic entities exhibit an extraordinary ability to withstand intense levels of ionizing radiation, which poses a significant threat to biological systems. When exposed to cosmic rays and other forms of radiation, they employ a suite of protective mechanisms to mitigate damage to their cellular structures. This resilience is largely attributed to a unique protein called D-sup, which plays a critical role in shielding DNA from radiation-induced harm. Research indicates that tardigrades can endure radiation doses that would be lethal for many other organisms, which raises intriguing questions regarding the limits of life in extreme environments. In testing scenarios, individuals have survived exposure to radiation levels up to 5,000 times greater than what a human can tolerate. This phenomenon is particularly relevant in the context of space exploration, where cosmic rays permeate the vacuum of space with unrelenting intensity. The implications for astrobiology are profound. If tardigrades can thrive in such hostile conditions, it opens possibilities for the existence of life on other celestial bodies exposed to similar radiation. Understanding these survival strategies may not only illuminate the tenacity of life on Earth, but also guide future missions into the depths of the cosmos, where the potential for discovering life may reside among the stars. Vacuum conditions present a formidable challenge for most organisms, yet tardigrades exhibit remarkable resilience in this environment. Surviving the vacuum of space requires an extraordinary suite of adaptations, allowing these microscopic travelers to endure extreme desiccation and radiation exposure. In a state known as cryptobiosis, metabolism is nearly halted, effectively suspending life processes. This transformation involves the expulsion of nearly all water from their bodies, creating a desiccated form that can withstand harsh conditions. Studies have revealed that tardigrades can endure vacuum for extended periods, showcasing their unique ability to protect cellular structures during exposure. Their proteins, particularly a group known as protective proteins, aid in stabilizing cellular membranes and preserving vital molecular integrity. This resilience enables them to survive not only in the absence of air but also under intense radiation and extreme temperatures. In experiments conducted in the vacuum of low Earth orbit, tardigrades have emerged unscathed after exposure to conditions that would obliterate most other life forms. Scientists are now exploring how these tiny organisms can inform astrobiological research, potentially revealing pathways for life to exist beyond Earth. Their capacity to endure the void serves as a reminder of life's remarkable adaptability, presenting new avenues for understanding the limits and possibilities of biology in the universe. The European Space Agency has conducted experiments to study the resilience of tardigrades in the harsh environment of space. These microscopic organisms were sent aboard the Photon M3 mission, where they were exposed to the vacuum and radiation of low Earth orbit. This endeavor sought to unravel the mechanisms that enable them to withstand such extreme conditions. Survivability tests revealed that tardigrades could endure prolonged exposure to space, remaining viable even after several days without water. The testing environment simulated the conditions of outer space, providing invaluable insights into how life can persist beyond Earth's atmosphere. The experiments observed the role of cryptobiosis, a state in which metabolism ceases and cellular structures are preserved. In addition to their resilience, the data collected highlights the potential for tardigrades to contribute to astrobiology. Their ability to survive radiation levels far exceeding those found on Earth raises questions about the resilience of life elsewhere in the cosmos. Understanding these mechanisms may provide clues to the origins of life and its possible existence on other celestial bodies. These findings underscore the significance of tardigrades as models for studying extremophiles. They offer a glimpse into the profound adaptability of life, challenging long-held perceptions of biological limits in the vast expanse of space. Trehalose serves a pivotal function in the resilience of tardigrades, acting as a protective agent during extreme environmental conditions. This sugar, found in various organisms, is particularly abundant in these microscopic travelers. It plays a crucial role in stabilizing proteins and cellular structures when dehydration occurs. When subjected to desiccation, trehalose effectively replaces water molecules, forming a glass-like state that preserves cellular integrity. In the absence of water, these creatures enter a cryptobiotic state, wherein metabolic processes nearly halt. Trehalose prevents the formation of ice crystals, which could otherwise puncture cellular membranes and lead to cell death. This mechanism has been observed in specimens that endure temperatures ranging from absolute zero to well above boiling point. The presence of trehalose enables them to resist radiation and desiccation, conditions that would be lethal for most life forms. As they encounter the vacuum of space, trehalose continues to play a vital role. The ability to withstand cosmic radiation and the harsh conditions of outer space illustrates the remarkable versatility of life. While their microscopic size belies their capabilities, these organisms reveal significant insights into survival strategies. New research continues to explore the potential applications of trehalose in biotechnology and medicine, extending its relevance beyond the realm of tardigrades. The resilience of tardigrades extends beyond their unique biochemical adaptations, revealing sophisticated DNA repair mechanisms. When exposed to extreme conditions, such as ultraviolet radiation or desiccation, they employ a remarkable suite of strategies to mend damaged genetic material. This region of research highlights the organism's ability to withstand environmental stresses that would be lethal to most life forms. Key among these mechanisms is the process of homologous recombination. It enables tardigrades to accurately repair double-strand breaks in their DNA, a common form of damage caused by radiation or other stresses. This precise repair pathway allows them to retain genomic integrity even after severe exposure. Alongside homologous recombination, they utilize non-homologous end joining, a more error-prone method that swiftly reconnects broken DNA ends when time is of the essence. The presence of unique proteins within these microscopic travelers further enhances their repair capabilities. Some of these proteins are believed to protect DNA from oxidative damage, while others play roles in regulating the repair processes. These adaptations underscore an evolutionary history finely tuned to survive conditions that would obliterate many other organisms. Understanding these DNA repair mechanisms may offer insights into enhancing stress resistance in other species, including crops and human cells. The study of tardigrades thus serves as a bridge between extremophilic biology and broader applications in science and medicine. Protective proteins play a crucial role in the survival of tardigrades under extreme conditions. These microscopic organisms possess unique proteins known as protective cuticle proteins, which form an essential barrier against desiccation, temperature fluctuations, and radiation. During the process of cryptobiosis, in which metabolism effectively halts, these proteins undergo significant structural transformations. This adaptation allows the organisms to withstand environments that would otherwise be lethal. By stabilizing cellular structures during periods of intense stress, protective proteins contribute to the resilience found in these tiny beings. They prevent the formation of ice crystals within cells during freezing conditions and mitigate damage from ultraviolet radiations. The mechanisms by which these proteins function are a subject of ongoing research, revealing insights into cellular protection and repair. In addition to protective cuticle proteins, tardigrades also produce a class of proteins called trehalose, which acts as a sugar that protects cellular integrity. This strategy enables them to retain water even in desiccated states while maintaining metabolic potential. The study of these proteins not only illuminates the biological intricacies of tardigrades, but also has implications for biopreservation techniques and synthetic biology. Understanding the composition and function of protective proteins can potentially lead to advancements in various fields, including medicine, agriculture, and biotechnology, where resilience is paramount. Many stronal adaptations play a crucial role in the survival of tardigrades. These remarkable organisms exhibit an extraordinary ability to endure conditions that would be lethal to most life forms. In extreme dehydration, they enter a state known as cryptobiosis, effectively suspending their metabolic processes. This transformation allows them to withstand severe droughts, extreme temperatures, and even radiation. Substantial research has documented their resilience against cold, with some species able to survive temperatures close to absolute zero. In contrast, others thrive in scorching environments, withstanding heat exceeding 100 degrees Celsius. Such versatility indicates a profound connection between their physiological makeup and the environments they inhabit. Microhabitats, often overlooked, serve as refuge for these creatures. Mosses, lichens, and leaf litter provide shelter, moisture, and nutrients. These regions act as miniature ecosystems, where tardigrades can find sustenance while being shielded from the harshness of the external environment. Tardigrades also demonstrate the ability to endure high levels of ionizing radiation, attributed to unique DNA repair mechanisms. This proficiency suggests that they could serve as models for understanding resilience in other organisms, potentially informing future research in fields such as astrobiology. The study of their adaptive strategies enriches our understanding of life's tenacity in extreme conditions across the universe. Tardigrades inhabit diverse environments, including the intricate ecosystems found within soil. These microscopic organisms coexist alongside a multitude of microbes, fungi, and larger soil-dwelling creatures. Their interactions within this complex community contribute significantly to soil health and nutrient cycling. Through their feeding habits, they influence microbial populations, sometimes enhancing the availability of nutrients for plant growth. The presence of tardigrades in soil can promote microbial diversity, fostering a more resilient ecosystem. They feed on bacteria, algae, and small protozoa, effectively regulating these populations and preventing any single species from dominating. This predation helps maintain a balance, ensuring that various microbial communities can thrive. Interactions between tardigrades and other soil microorganisms are also notable. Some microbes may form symbiotic relationships with these resilient travelers, providing them with essential nutrients while benefiting from the tardigrades' feeding activities. In turn, the physical activity of tardigrades contributes to the aeration of the soil, enhancing water retention and promoting overall soil structure. Research into these interactions reveals a delicate web of relationships, illustrating how life, no matter how small, plays a crucial role in the stability and functionality of ecosystems. The resilience of tardigrades, combined with their ecological contributions, underscores their importance in understanding life's adaptability within microbial landscapes. Interconnected ecosystems reveal the intricate relationships that define survival. Tardigrades often engage in symbiotic interactions with other microorganisms, enhancing their ecological roles. Within damp mosses and lichen, these minute creatures coexist alongside bacteria, fungi, and protozoa, forming a complex web of life. Such relationships provide mutual benefits, with tardigrades consuming bacterial populations while contributing to nutrient cycling through their feeding habits. In these environments, the presence of tardigrades influences microbial community dynamics. By grazing on bacteria, they regulate microbial abundance, ensuring that no single species dominates. This balance is crucial for maintaining the health of their habitats. The interplay between these organisms illustrates the importance of biodiversity within even the smallest ecosystems. Additionally, some tardigrades are known to harbor specific symbiotic bacteria within their bodies. These bacteria can enhance the tardigrade's resilience, especially in extreme conditions. The partnership helps shield them from environmental stresses, such as desiccation and radiation. In return, the bacteria benefit from the stable environment provided by their host. These relationships emphasize the complexity of life, demonstrating that survival often hinges on cooperation rather than competition. The study of these interactions offers insights not only into the adaptability of tardigrades, but also into the broader mechanisms that sustain life across diverse ecosystems. These microscopic organisms occupy a unique niche within various ecosystems, serving as both prey and predators. Their presence in soil, leaf litter, and aquatic environments contributes to a complex food web where they influence population dynamics. As primary consumers, tardigrades feed on microorganisms such as algae, fungi, and bacteria. This feeding behavior helps regulate microbial populations and facilitates nutrient cycling, enhancing soil fertility and ecosystem health. Predatory tardigrades engage in a different role, preying upon smaller microorganisms and maintaining balance within their habitats. Their ability to thrive in extreme conditions means they can persist in environments where other organisms may fail, further solidifying their role as resilient components of the food web. In nutrient-poor environments, their presence can be particularly significant, as they contribute to the stability of microbial communities, allowing for the establishment of diverse life forms. The interactions of tardigrades with other organisms create a web of dependencies that underpins ecosystem functionality. Their ability to tolerate extremes also underscores the potential for survival strategies that could inform our understanding of life's resilience. Research continues to reveal the intricate relationships they maintain within these ecosystems, shedding light on the essential roles they play and how these roles may influence broader ecological processes. The study of these relationships is crucial in understanding the complexities of life on Earth. Tardigrades play a pivotal role in the ecosystems they inhabit, contributing significantly to the overall biodiversity. This microscopic group can be found in diverse environments, from the depths of the ocean to the highest mountain peaks. Their presence in varied habitats enhances the resilience of these ecosystems, as they interact with numerous organisms, including bacteria, fungi, and plants. In soil ecosystems, they contribute to nutrient cycling, aiding in the breakdown of organic materials. As they consume microorganisms, they facilitate the transfer of energy through food webs, supporting the growth of larger organisms. Their ability to thrive in extreme conditions allows them to act as indicators for environmental changes, providing insights into ecosystem health. Tardigrades also demonstrate remarkable adaptability, allowing them to occupy ecological niches that may otherwise remain uninhabitable. This adaptability fosters genetic diversity, which is essential for the resilience of ecosystems facing climate change and other anthropogenic pressures. In addition, the study of these organisms has implications for biotechnology and astrobiology. Understanding their survival mechanisms may lead to advancements in medicine and help scientists search for life beyond Earth. The ecological importance of tardigrades extends far beyond their minuscule size, highlighting their essential role in maintaining the delicate balance of life on this planet. Tardigrades serve as valuable bioindicators, reflecting the health of their environments with remarkable precision. These resilient organisms inhabit diverse ecosystems, from the depths of the oceans to the highest mountain ranges, making them integral to ecological monitoring. Their presence or absence often signals changes in environmental conditions such as pollution levels, habitat degradation, or climate shifts. This adaptability allows them to thrive in extreme situations, yet it also makes them sensitive to subtle alterations in their habitats. Research indicates that certain species respond distinctly to specific pollutants, providing a clear signal of ecological stress. When environmental quality declines, tardigrade populations often decrease or exhibit physiological changes, making them reliable markers for scientists assessing ecosystem health. Their ability to withstand extreme conditions, including desiccation and radiation, does not detract from their vulnerability to chemical pollutants and habitat disturbances. In laboratory settings, scientists study tardigrades to understand the impacts of various stressors on biodiversity and ecosystem functionality. This research contributes to broader ecological assessments, supporting conservation efforts aimed at preserving fragile environments. Recognizing the significance of these microscopic travelers reinforces the understanding that even the smallest life forms can have profound implications for environmental monitoring and the preservation of biodiversity on Earth. Genetic studies of tardigrades reveal remarkable adaptations that enable survival in extreme environments. The sequencing of their genomes provides insights into the unique molecular mechanisms that confer resilience against factors such as desiccation, radiation, and extreme temperatures. Specific genes associated with damage repair and protein stabilization have been identified, highlighting the evolutionary pathways that support such robustness. This region of research focuses on the presence of protective proteins known as trehalose, which play a critical role in cellular preservation during dehydration. By stabilizing cellular structures, these proteins enable tardigrades to endure conditions that would be fatal for most organisms. Additionally, the expression of particular genes allows for rapid recovery and rehydration upon encountering water again. Investigations into genetic diversity among tardigrade species further enrich the understanding of their adaptability. Varied genomic adaptations across different habitats illustrate the evolutionary responses to environmental pressures. These findings contribute to broader discussions about the resilience of life forms in extraterrestrial environments, particularly in astrobiology. Insights from tardigrade genomes not only enhance knowledge of their survival strategies, but also prompt inquiries into the potential applications of these mechanisms in biotechnology. Understanding the genetic foundations of such extraordinary survival enhances the broader narrative of life and resilience on our planet and beyond. This lineage reveals a fascinating journey through time, tracing back to the Cambrian period over 500 million years ago, The earliest ancestors of tardigrades thrived in a diverse array of environments, adapting remarkably to the challenges presented by their surroundings. Morphological studies indicate that they share a common ancestor with arthropods, which suggests a deep evolutionary connection to some of the most successful organisms on Earth. Over eons, these microscopic entities developed unique characteristics, including a robust cuticle that provides protection from desiccation, extreme temperatures, and radiation. Such adaptations positioned them as resilient survivors within ecosystems ranging from the depths of the ocean to the highest mountains. Genetic analyses reveal that their evolutionary path diverged from that of other invertebrates, allowing them to inhabit niches where few others could endure. This evolutionary history underscores the significance of tardigrades as a model for studying resilience and adaptability. Their capacity to enter a cryptobiotic state, a form of suspended animation, exemplifies the intricate mechanisms life employs to withstand adverse conditions. As research continues, insights into their ancestry may illuminate not only the resilience of life on Earth, but also the potential for existence beyond our planet, where the harshness of the cosmos could mirror the challenges faced by their ancient relatives. Phylogenetics reveals intricate evolutionary relationships among tardigrades, shedding light on their diversification and adaptation strategies. Molecular analyses indicate that these microscopic organisms share a common ancestry with arthropods and nematodes, suggesting a complex evolutionary history. Genetic sequencing has provided evidence for the existence of distinct lineages within the tardigrade phylum, illustrating their ability to thrive in diverse environments. The evolutionary pathways taken by these resilient creatures have allowed them to colonize ecosystems ranging from the deep sea to the highest mountain ranges. Their ability to undergo cryptobiosis enables survival in extreme conditions, a trait likely inherited from ancestral forms that faced similar environmental pressures. This phenomenon of desiccation tolerance and metabolic shutdown reflects their long-standing relationship with the planet's fluctuating climates. Phylogenetic studies have also uncovered the role of horizontal gene transfer, a process through which tardigrades may acquire genetic material from other organisms, enhancing their adaptability. Such mechanisms could play a crucial role in their survival as they encounter new challenges in changing environments. Understanding these evolutionary connections not only informs the biology of tardigrades, but also provides insights into the broader patterns of life on Earth, emphasizing the interconnectedness of all living organisms in the relentless march of evolution. Tardigrades exhibit remarkable resilience, yet they are not the only extremophiles thriving in harsh environments. Other microorganisms, such as bacteria and archaea, also demonstrate extraordinary survival strategies. These organisms inhabit extreme conditions, from scorching hot springs to deep-sea hydrothermal vents, showcasing diverse adaptations. While tardigrades can endure extreme desiccation and radiation, certain bacteria can withstand intense temperatures, surviving in environments exceeding 120 degrees Celsius. In stark contrast, some archaea thrive in highly saline environments where salt concentrations render most life forms unviable. These microorganisms employ unique biochemical mechanisms to maintain cellular integrity under osmotic stress, a challenge that would render tardigrades vulnerable. The ability of tardigrades to enter a state of cryptobiosis allows them to suspend metabolic processes entirely, a strategy not commonly shared among all extremophiles. These microorganisms also differ in their evolutionary paths. Tardigrades belong to a distinct lineage, exhibiting traits that are not present in other extremophiles. Their unique cuticle and specialized cellular structures provide a protective barrier, enabling them to withstand conditions that would otherwise be lethal. This region of study highlights the vast array of survival strategies across the microbial world. revealing the intricate web of life and the endless possibilities of adaptation in the face of adversity. The role of tardigrades in astrobiology underscores their potential as key players in understanding life's resilience beyond Earth. These microscopic organisms exhibit an extraordinary ability to withstand extreme conditions, including intense radiation, extreme temperatures, and even the vacuum of space. Their remarkable survival mechanisms, such as cryptobiosis, allow them to enter a state of suspended animation, effectively halting their metabolic processes. Research indicates that tardigrades possess unique proteins that protect their cellular structures from damage during desiccation and radiation exposure. This capability raises important questions about the possibility of life existing on other celestial bodies, such as Mars or the icy moons of Jupiter and Saturn. The presence of water and organic compounds in these environments suggests that similar mechanisms might allow life, even in its most primitive forms, to thrive. Experiments conducted in low Earth orbit have demonstrated that tardigrades can endure the harsh conditions of space travel, with some individuals surviving exposure to cosmic radiation. This resilience implies that if life does exist elsewhere in the universe, it may share certain characteristics with these tiny organisms. Understanding the biological limits of survival can provide valuable insights into the search for extraterrestrial life and the conditions necessary for life to emerge and persist. The study of tardigrades continues to illuminate the broader implications of life's adaptability across the cosmos. Research into these resilient organisms indicates a promising future for understanding extremophiles in various environments. Scientists are focusing on the genetic makeup of tardigrades, aiming to unlock the secrets behind their extraordinary survival mechanisms. Their unique proteins, particularly those involved in desiccation and radiation resistance, are of significant interest. This knowledge may pave the way for advancements in biotechnology, potentially leading to innovations in crop resilience and preservation techniques. Current trends emphasize the application of cutting-edge genomic sequencing technologies as researchers decode the genome of different tardigrade species. Comparisons with other organisms may reveal evolutionary pathways that highlight adaptations to extreme conditions. This comparative analysis could deepen insights into cellular repair processes and stress responses. Collaborative efforts among international scientific teams are increasing, fostering a multidimensional approach to tardigrade research. Interdisciplinary studies often merge microbiology, astrobiology, and environmental sciences, expanding the context of their findings. The exploration of habitats, such as deep-sea trenches and polar ice, continues to provide valuable data on the ecological roles of these tiny creatures. Investigating the potential for tardigrades in astrobiology represents a crucial aspect of future research. Understanding their survival in space conditions could inform strategies for detecting extraterrestrial life and the possibilities for human exploration beyond Earth. The resilience of tardigrades extends beyond the cosmic void. In an era marked by climate change, these microscopic entities reveal insights into ecological dynamics. Their ability to withstand extreme conditions raises questions about their role in shifting environments. As temperatures rise and ecosystems undergo significant transformations, the adaptability of tardigrades may serve as an indicator of broader biological responses. Populations of these organisms can be found in diverse habitats, from the frozen tundras to the deepest ocean trenches. As climates become increasingly volatile, alterations in their habitats may influence their survival and distribution. Increased temperatures and fluctuating moisture levels could affect their reproductive cycles and overall biodiversity. Research indicates that tardigrades' capacity to enter a cryptobiotic state allows them to endure periods of desiccation and extreme temperatures. This feature may provide a unique advantage in rapidly changing environments, as they can temporarily halt their metabolic processes. Such adaptability highlights their potential as bioindicators for assessing ecosystem health amid climate stresses. Ongoing studies explore the implications of climate change on tardigrade populations, aiming to understand how these organisms may inform our comprehension of ecological resilience, the potential consequences of environmental changes on their habitats. The role of these microscopic travelers in soil health and restoration is intricate and vital. Tardigrades contribute to the soil ecosystem by participating in the decomposition process. Their feeding habits facilitate the breakdown of organic matter, which enhances nutrient cycling. This activity supports the growth of both microbial communities and plant life, creating a balanced environment. In addition to decomposition, these resilient organisms also serve as indicators of soil health. Their presence often signifies a thriving ecosystem, as they require a stable environment to flourish. Monitoring their populations can provide insights into soil conditions and the impact of environmental changes. Will they of restoration? Tardigrades play a pivotal role when soils are disturbed or degraded. The introduction of these creatures can aid in the re-establishment of ecological balance. Their resilience allows them to survive in extreme conditions, making them essential in reclaiming damaged habitats. They can withstand desiccation and radiative events, providing a unique advantage in areas undergoing rehabilitation. Research into their interactions within soil matrices continues to reveal their importance in maintaining ecological integrity. By studying these organisms, scientists gain insights that influence conservation efforts. Their contributions to soil health exemplify the interconnectedness of life, showcasing how even the smallest beings can impact broader environmental processes. The influence of these resilient organisms extends beyond the confines of scientific research. Tardigrades have captured the imagination of artists and creators, embodying themes of endurance and survival in various forms of expression. Their unique ability to withstand extreme conditions has inspired numerous artistic interpretations, from intricate illustrations to larger-than-life sculptures. This fascination is evident in popular culture, where these minute beings often symbolize tenacity and hope. They appear in literature and cinema, serving as metaphors for resilience in the face of adversity. In graphic novels and animated series, the depiction of these creatures as adventurous survivors invites contemplation on the fragility of life and the strength found within even the smallest entities. Exhibitions showcasing the microscopic world frequently highlight the tardigrade, emphasizing its role as a bridge between science and art. This blending of disciplines encourages viewers to reflect on the complexities of existence and the beauty inherent in the natural world. As the interest in such organisms continues to grow, so does their significance in human culture, illustrating the ways in which scientific discoveries can transcend their original context. The legacy of tardigrades is thus not only one of biological marvel, but also of cultural inspiration, fostering a deeper appreciation for the interconnectedness of life across various domains. This fascination has permeated popular media, leading to a surge in representations of these microscopic entities across various platforms. Documentaries and educational programs often highlight their resilience, showcasing footage that captures their unique survival strategies. This visibility has sparked curiosity among audiences, transforming them into symbols of endurance and adaptability. Children's books featuring tardigrades have emerged, Designed to introduce young minds to the wonders of the microscopic world. Illustrations depict these creatures in vibrant colors, enhancing their appeal while conveying scientific principles. Television shows, including animated series, have incorporated them as characters, further embedding their image in contemporary culture. Merchandise related to these organisms has proliferated, with toys, clothing, and accessories bearing their likeness. This trend illustrates how the scientific narrative can inspire creativity, allowing these organisms to become icons of resilience in everyday life. Social media platforms have also played a significant role in disseminating information and fostering communities centered around these tiny survivors, facilitating discussions among enthusiasts and scientists alike. Artistic interpretations often portray tardigrades in whimsical contexts, serving as reminders of nature's wonders. Such representations not only celebrate their biological significance, but also enhance public engagement with science, prompting deeper inquiries into the natural world. The influence of these organisms thus extends beyond academia, enriching popular culture and scientific literacy. Tardigrades offer unique educational opportunities, serving as effective tools for engaging students in the sciences. Their resilience, ability to withstand extreme conditions, and fascinating biology stimulate curiosity, prompting inquiries into microbiology, environmental science, and astrobiology. Classroom experiments involving these microscopic organisms allow students to observe remarkable properties, such as cryptobiosis, where they enter a dormant state to endure desiccation or extreme temperatures. Various resources are available to educators, enabling the incorporation of tardigrades into diverse curricula. Textbooks, online platforms, and laboratory kits provide detailed protocols for cultivation and observation. These materials foster interactive learning experiences, allowing students to collect samples from mosses or lichens, subsequently examining them under microscopes. The exploration of tardigrades can ignite discussions about evolution and the adaptability of life in harsh environments, linking concepts from genetics to ecological resilience. In addition to hands-on activities, multimedia presentations can enhance understanding. Documentaries and educational videos illustrate the habitats and survival strategies of tardigrades, further enriching the learning experience. Collaborations between scientists and educators yield workshops and public outreach programs that promote awareness of these organisms and their significance in understanding life's potential beyond Earth. As educational tools, tardigrades bridge the gap between complex scientific concepts and accessible learning, inspiring future generations to explore the wonders of the microscopic world. Microscopy techniques provide the means to observe tardigrades in their natural habitats and under various conditions. Optical microscopy allows researchers to view these resilient organisms in their active state, revealing their unique morphology and locomotion patterns. Light microscopy captures the intricate details of their body structure, including the segmented appearance and specialized appendages used for movement. Fluorescence microscopy enhances the observation of tardigrades by utilizing specific dyes that bind to cellular components, allowing scientists to visualize internal structures and metabolic processes. This method can highlight the organism's response to environmental stresses, offering insights into their extraordinary survival mechanisms. Electron microscopy presents an even more detailed perspective, enabling visualization at the nanometer scale. This technique exposes the complexity of tardigrade anatomy, providing a closer look at their protective cuticle and specialized cells that assist in desiccation and radiation resistance. Confocal microscopy further refines imaging capabilities, allowing for three-dimensional reconstructions of tardigrades. This method reveals how they navigate through their environments, providing a clearer understanding of their ecological roles. These varied microscopy techniques contribute to a growing body of knowledge about tardigrades. Each method offers unique insights, enriching our comprehension of these remarkable organisms and their adaptability to extreme conditions in the microscopic ecosystem. Field studies reveal a vast array of habitats that harbor tardigrades, from the icy realms of polar regions to the sun-drenched surfaces of mosses in temperate forests. Researchers meticulously collect samples, often using a fine brush or a gentle vacuum, ensuring minimal disturbance to these fragile ecosystems. These environments act as living laboratories, showcasing the resilience of life in the face of harsh conditions. Microscopic examinations follow, revealing the physicality of these minute creatures, segmented bodies, clawed limbs, and intricate mouthparts designed for a diet primarily composed of plant cells and microorganisms. Observations highlight their capacity for cryptobiosis, a dormant state allowing survival during extreme desiccation, intense radiation, and even the vacuum of space. The diversity of tardigrades extends to their genetic makeup, identified through advanced genomic techniques. Each species, adapted to its unique niche, contributes to a broader understanding of evolutionary processes. In laboratory settings, scientists simulate environmental extremes, prompting these organisms to enter their cryptobiotic state. This research fosters a deeper appreciation for the tenacity of life, showcasing the interconnectivity of ecosystems. As studies progress, the implications of tardigrade resilience extend beyond mere curiosity, influencing fields such as astrobiology and biotechnology, where their survival mechanisms may inspire innovations for human use. Laboratory culturing presents a unique approach to studying tardigrades in controlled environments. Techniques have evolved to efficiently propagate these microscopic organisms, allowing for extensive research on their biology and resilience. Cultivation often begins with the careful selection of habitats, mimicking the natural environments where they thrive. Moist substrates, typically consisting of moss or lichen, provide the necessary nutrients and moisture essential for their growth. Temperature control is vital, as these organisms exhibit optimal activity within specific ranges. Maintaining consistent humidity levels ensures that the environment remains suitable for reproduction. Light exposure is minimized, as excessive illumination can be detrimental. Researchers utilize darkened chambers to simulate their natural habitats, where light is often scarce. Feeding protocols are established, leveraging a diet of algae and microorganisms to support healthy populations. Observations of feeding behaviors contribute to a deeper understanding of their ecological roles. The process of propagation often involves isolating individuals to monitor reproduction, yielding insights into their life cycles and genetic diversity. In addition, cryopreservation techniques are employed to store tardigrades for future study, preserving genetic material and ensuring that populations remain viable for years. These practices are crucial not only for research, but also for potential applications in space exploration, where the ability to withstand extreme conditions is paramount. And engaging the public in tardigrade research opens new avenues for scientific discovery. Community involvement fosters a deeper understanding of these remarkable organisms and their resilience. Citizen scientists, equipped with simple tools, can collect samples from diverse environments, contributing valuable data to ongoing studies. This participation not only enriches research, but also inspires curiosity about the microscopic world. Programs aimed at encouraging public engagement often focus on local ecosystems. These environments, ranging from moss-covered rocks to the surface of lichens, serve as prime habitats for tardigrades. Through hands-on activities, individuals learn to identify and collect specimens, enhancing their observational skills while participating in scientific inquiry. The collaboration between trained researchers and enthusiastic amateurs exemplifies the synergy that can arise from such partnerships. Data gathered by citizen scientists can reveal patterns and trends in tardigrade populations, providing insight into their ecological roles. The findings may also highlight the impact of environmental changes on these resilient creatures. Researchers can analyze variations in distribution and adaptation, further expanding knowledge about the limits of life on Earth. This initiative embodies a broader movement towards inclusivity in science, where the contributions of all individuals are recognized. As interest in terrestrial and extraterrestrial life continues to grow, this collective effort becomes increasingly significant in understanding life's tenacity in the universe. In the pursuit of understanding these remarkable organisms, the preservation of their habitats emerges as a crucial endeavor. Tardigrades occupy extreme environments, from the depths of the ocean floor to the highest mountain ranges, each unique setting contributing to their resilience. These regions, often overlooked, are home to complex ecosystems where the balance of life is delicate. Protecting them is not merely an act of conservation, it is fundamental to maintaining biodiversity. Human activity poses significant threats to these habitats, with pollution, climate change, and habitat destruction leading to the degradation of the environments where tardigrades thrive. In urban settings, soil compaction and chemical runoff can drastically alter the microhabitats essential for their survival. Conservation efforts must focus on establishing protected areas, promoting sustainable practices, and raising awareness about the importance of these microscopic life forms. Scientific research plays a pivotal role in this conservation effort by studying the specific conditions that allow tardigrades to flourish. It becomes possible to implement targeted measures aimed at safeguarding their environments. Restoration projects that rehabilitate damaged ecosystems can also support the recovery of these resilient travelers, fostering a deeper appreciation for the intricate relationships within these ecosystems can lead to more effective conservation strategies, thereby ensuring that tardigrades, alongside countless other species, continue to thrive on Earth. Research into tardigrades raises significant ethical considerations surrounding the responsibilities of scientists and conservationists. Understanding the survival mechanisms of these microscopic organisms not only enhances knowledge of resilience, but also opens avenues for biotechnological applications. This exploration must be approached with caution, as the manipulation of such life forms carries potential consequences that reach far beyond the laboratory. The genetic material of tardigrades, known for its remarkable stability, invites scrutiny regarding the ethical implications of bioengineering. Considerations must be made about the potential impacts on ecosystems if these organisms were introduced into new environments or altered for human use. The balance between scientific advancement and ecological integrity becomes paramount, demanding rigorous assessments of risks and benefits. Public engagement in these discussions is essential, fostering transparency and understanding of the implications of research. Ethical frameworks guiding research practices must prioritize the welfare of not only tardigrades, but the interconnected web of life within which they exist. This region of inquiry invites collaboration among scientists, ethicists, and policymakers, ensuring that advancements in knowledge remain aligned with the principles of stewardship and conservation. A thoughtful approach to the study of these resilient travelers can contribute to a more sustainable relationship between humanity and the natural world, safeguarding the delicate balance of ecosystems on Earth. Future applications of tardigrade proteins hold significant promise for biotechnology. These proteins, known for their remarkable stability and resistance to extreme conditions, may unlock new avenues for innovation. Their ability to protect cellular structures from dehydration could inform the development of preservation techniques for biological materials, including tissues and organs. This potential extends to enhancing the longevity of food products and pharmaceuticals, ensuring they remain viable in adverse environments. Research indicates that the proteins also exhibit antifreeze properties, which may lead to advancements in cryopreservation. Such techniques could revolutionize organ transplantation, allowing for longer storage times and improved success rates in transplants. The implications for medicine are profound, providing solutions to the challenges posed by limited organ availability. In agricultural settings, these proteins might be utilized to engineer crops with heightened resistance to drought and other stresses. This could contribute to food security as global climates continue to change, ensuring that essential crops thrive in increasingly hostile conditions. Furthermore, the exploration of tardigrades in the context of astrobiology offers insights into how life might exist beyond Earth. Understanding their adaptations could guide the search for extraterrestrial life forms and inform future missions aimed at exploring other planets. The ongoing study of these microscopic travelers reveals a world of possibilities, bridging the gap between the known and the unknown. Applications of tardigrades extend into the realm of pharmaceutical development. Their remarkable resilience offers insights into biostability, crucial for the creation of effective therapeutics. These organisms can withstand extreme temperatures, pressures, and radiation, suggesting potential pathways for preserving vaccines and biologics under challenging conditions. The ability to enter a cryptobiotic state, essentially suspending metabolic activity, presents opportunities for developing novel preservation techniques. Research indicates that substances found within tardigrades, such as protective proteins and sugars, may enhance the stability of fragile pharmaceutical compounds. These molecules could be pivotal in formulating medicines that retain efficacy over extended periods and under variable storage conditions. By harnessing the natural mechanisms employed by these organisms, scientists aim to improve the shelf life and delivery of essential drugs. In addition to preservation, the unique cellular mechanisms of tardigrades offer promising avenues in gene therapy. Their genetic material provides a reference point for understanding stress responses, potentially guiding the development of therapies targeting genetic disorders. By observing how they repair cellular damage, researchers can glean insights that may enhance the efficacy of treatments in human cells. This exploration into the applications of tardigrades demonstrates the broader implications of their survival strategies, highlighting their significance beyond mere curiosity in the microscopic world. Exploration into the role of tardigrades extends into astrobiological missions, where their resilience serves as a focal point for understanding life's potential beyond Earth. These organisms exhibit an extraordinary capacity to withstand extreme conditions, including intense radiation, extreme temperatures, and complete desiccation. Such traits position them as ideal candidates for experiments designed to assess the viability of life on other celestial bodies. In controlled environments, researchers have subjected tardigrades to conditions simulating those found on Mars and the icy moons of Jupiter and Saturn. The survival rates observed under these harsh circumstances provide crucial data regarding the limits of life. This information is essential for evaluating the likelihood of encountering extraterrestrial organisms, as well as for informing the design of life support systems for future human exploration. In addition to their resilience, tardigrades' ability to enter a state of cryptobiosis allows them to endure prolonged periods without water or nutrients. This characteristic is of particular interest when considering long-duration space missions or the potential for life in environments with sporadic availability of resources. These organisms exemplify the tenacity of life, offering a window into the adaptability required for survival in the cosmos. The ongoing study of these microscopic travelers continues to enrich our understanding of life's resilience and its potential to thrive beyond our planet. Tardigrades possess remarkable adaptations that allow them to endure extreme environments, including the vacuum of space. This resilience raises intriguing questions about the possibilities of life beyond Earth. Their ability to withstand radiation, extreme temperatures, and prolonged desiccation presents a model for understanding how life might exist on other celestial bodies. Research into these organisms has inspired scientists to explore environments like the icy moons of Jupiter and Saturn, where conditions may resemble those in which tardigrades thrive. In the search for extraterrestrial life, analyzing the conditions that support tardigrades can yield insights into the potential for similar organisms elsewhere. This line of inquiry extends to the exploration of Mars, where evidence suggests past water presence and potential habitability. The presence of extremophiles, such as tardigrades, provides a framework for assessing the viability of life in harsh environments. The examination of these microscopic beings also enhances the interpretation of findings from astrobiological missions. As spacecraft venture into unexplored regions, understanding how life can endure in extreme conditions informs the search for biosignatures. Discoveries of organic compounds or microbial activity in extraterrestrial soil could parallel the survival mechanisms seen in tardigrades. Thus, the study of these resilient organisms not only illuminates the possibilities for life on other worlds, but also emphasizes the interconnectedness of life throughout the cosmos. Fascination with tardigrades has permeated popular science, captivating audiences worldwide. Documentaries and articles highlight their extraordinary abilities, often portraying them as near-mythical survivors. This representation serves to bridge the gap between complex scientific concepts and public understanding. By emphasizing their resilience, it invites a broader appreciation for the delicate balance of life and the conditions that sustain it. Tardigrades are frequently featured in educational programs where their survival strategies are depicted as remarkable adaptations to extreme environments. High-definition imagery reveals their unique morphology, showcasing how their bodies can withstand temperature fluctuations, radiation, and even the vacuum of space. These visual representations enhance engagement, making the science accessible to a diverse audience. In the realm of literature, the portrayal of these organisms often transcends mere biological interest. Authors explore the philosophical implications of their existence, considering questions of life's tenacity and the potential for extraterrestrial forms. This narrative invites contemplation about humanity's place in the universe. Scientific research continues to expand, with studies delving into the genetic mechanisms that underpin tardigrades' survival. This exploration not only enriches our understanding of evolution, but also inspires innovative applications in biotechnology and medicine. The allure of these microscopic travelers continues to grow, bridging scientific inquiry with the human experience. Concentrations of microplastics infiltrate terrestrial and aquatic ecosystems, prompting concerns for various organisms. Tardigrades, known for their remarkable resilience, face potential threats from these synthetic particles. Research indicates that ingestion of microplastics can disrupt the digestive systems of these tiny creatures, leading to detrimental health effects. The incorporation of these foreign substances into their bodies raises questions about their long-term survival. This region, once teeming with diverse microbial life, now sees alterations in its ecological balance. Tardigrades consume organic matter, but the presence of microplastics may interfere with nutrient absorption. The impact on reproductive success and population dynamics remains an area of ongoing investigations. Studies suggest that exposure to microplastics can also lead to increased mortality rates, further threatening their populations. Environmental stresses, such as rising temperatures and habitat degradation, compound these challenges. The resilience of tardigrades has been well documented, but the cumulative effects of microplastics may prove to be an unprecedented obstacle. Exploring the interplay between these microscopic travelers and their increasingly polluted environments reveals insights into broader ecological implications. Understanding the fate of these organisms can illuminate the consequences of human activity on biodiversity, offering a lens through which to examine the health of our ecosystems. The resilience and vulnerability of tardigrades encapsulate the delicate balance within nature's intricate web. Climate change poses significant challenges to ecosystems worldwide, yet tardigrades exhibit remarkable adaptability in the face of such shifts. Found in diverse environments, these microscopic entities thrive in conditions ranging from extreme heat to intense cold. Their ability to enter a cryptobiotic state enables them to withstand desiccation and high levels of radiation, ensuring survival even when faced with drastic alterations in their habitat. This region, characterized by fluctuating temperatures and erratic moisture levels, provides a unique laboratory for observing the effects of environmental change. Tardigrades can endure desiccation for years, reviving when conditions become favorable. This resilience underlines their potential role as bioindicators, offering insights into ecosystem health and the impacts of climate variability. In the soil of forests, tundras, and even urban areas, these organisms contribute to nutrient cycling, demonstrating their ecological significance. Their presence often signals the ability of ecosystems to adapt or recover from disturbances. As climate patterns become increasingly erratic, studying their mechanisms of survival presents opportunities to enhance our understanding of resilience. These microscopic travelers serve as a testament to life's enduring capabilities. Their existence reinforces the idea that adaptability is crucial in the face of environmental challenges, providing a glimmer of hope amid the uncertainties of a changing world. The impact of urbanization on tardigrade habitats poses significant questions regarding their resilience. As cities expand, natural environments are increasingly replaced by concrete and asphalt. This transformation alters the moisture levels and soil composition essential for tardigrades. The intricate balance of their ecosystems becomes disrupted. In urban settings, soil often experiences compaction, limiting the air pockets required for these microorganisms to thrive. Additionally, pollution introduces contaminants that can affect the delicate biochemical processes within tardigrades. The presence of heavy metals and chemicals can lead to increased mortality rates. Despite their robust nature, these microscopic travelers are not immune to the consequences of human activity. The fragmentation of their habitats creates isolated pockets, leading to reduced genetic diversity. In contrast, Some tardigrades exhibit a remarkable capacity to endure extreme conditions. This adaptability may allow certain populations to persist in urban environments, utilizing new and unexpected niches. They can inhabit mosses and lichens that grow on urban structures, finding refuge in the crevices of buildings and sidewalks. This resilience suggests a potential for survival even in altered habitats, highlighting the complexity of life amid anthropogenic changes. Research continues to explore the balance between urban development and the survival of tardigrades, emphasizing the importance of conservation in an increasingly urbanized world. These diminutive organisms inhabit diverse environments, ranging from the deep sea to the highest mountain ranges, adapting to extreme conditions through remarkable resilience. Microbiomes play a significant role in their survival, forming intricate relationships with various microorganisms. In these interconnected ecosystems, tardigrades benefit from the diverse microbial communities that coexist within their habitats. The presence of bacteria, fungi, and other microorganisms influences the nutritional availability for tardigrades. These tiny travelers exhibit an ability to incorporate beneficial microbes into their own systems, enhancing their metabolic processes. As they traverse through moist soil or lichen, the interactions with these microorganisms enhance their capacity to withstand harsh conditions, including desiccation and radiation. This region, characterized by its unique microbial diversity, serves as a microcosm of ecological balance. The interplay between tardigrades and their associated microbiomes reflects a complex web of life, where each organism contributes to the overall stability and resilience of the ecosystem. When environmental changes occur, such as shifts in temperature or moisture levels, these communities adapt, showcasing the remarkable plasticity of life. Understanding these interactions not only sheds light on tardigrades' survival strategies, but also emphasizes the crucial role of microbiomes in maintaining ecosystem health. The resilience of these microscopic adventurers highlights the intricate connections that sustain life in a multitude of environments. This region of Antarctica serves as a unique laboratory for studying the adaptability of tardigrades. In the harsh climate, where temperatures plunge far below freezing and the sun remains absent for months, these creatures demonstrate remarkable survival strategies. They inhabit the ice and moss, thriving in conditions that would be lethal to most other forms of life. Research indicates that tardigrades enter a state of cryptobiosis when faced with extreme conditions. This process allows them to suspend their metabolism, effectively pausing their biological functions for extended periods. In this state, they can withstand temperatures ranging from just above absolute zero to well above 100 degrees Celsius. The ability to endure extreme desiccation further exemplifies their tenacity, with some specimens capable of surviving without water for decades. In Antarctic studies, scientists have discovered tardigrades in glacial ice and subglacial lakes, revealing their capacity to colonize some of the most inhospitable environments on Earth. The presence of these organisms in such locations raises intriguing questions about the potential for life on celestial bodies, such as Europa or Enceladus, where conditions may mirror these extreme habitats. Understanding their survival mechanisms not only expands knowledge of life on Earth, but also enhances the search for extraterrestrial life in our solar system. Longevity in tardigrades presents an intriguing frontier in aging research. These microscopic organisms exhibit remarkable resilience, allowing them to endure extreme conditions including desiccation, radiation, and temperature fluctuations. Their ability to enter a state of cryptobiosis effectively halts metabolic processes, enabling them to survive for decades without water or nutrients. This dormant state raises questions about the biological mechanisms underlying aging and preservation. Research has identified specific proteins, such as trehalose, which play a crucial role in stabilizing cellular structures during desiccation. In the presence of water, these proteins rehydrate the tardigrade, allowing it to resume its metabolic functions. The implications of such mechanisms extend beyond the species itself. They may offer insights into preserving human cells and tissues for medical applications. Genetic studies reveal that tardigrades possess unique genes that contribute to their extraordinary longevity. These genes may regulate stress responses and cellular repair, fundamental processes that govern aging. Investigating these genetic factors could illuminate paths for extending lifespans in other organisms, including humans. The combination of survival strategies and genetic resilience positions tardigrades as a focal point in the quest to understand aging. Their enigmatic longevity invites further exploration, potentially transforming current paradigms in biological science and longevity studies, reshaping views on the limits of life. The remarkable resilience of tardigrades extends beyond their longevity. It is intricately linked to their unique biological composition. These tiny organisms possess a suite of proteins and sugars that contribute to their extraordinary resistance against extreme conditions. When faced with desiccation, they enter a state known as cryptobiosis, wherein metabolic processes nearly cease, allowing them to withstand dehydration levels that would be lethal to most life forms. This ability to endure harsh environments is attributed in part to trehalose, a sugar that protects cellular structures during drying. It stabilizes proteins and membranes, effectively preserving cellular integrity in the absence of water. The presence of protective proteins, such as the damage suppressor protein, also plays a critical role, shielding cellular components from damage caused by radiation and other stressors. Y2A that tardigrades can survive conditions that mimic the vacuum of space, exposing them to cosmic radiation and extreme temperatures. These adaptations provide insights into potential applications for biotechnology and pharmaceuticals, particularly in developing new methods for preserving biological materials. Understanding the chemistry behind their resilience could lead to advances in space exploration, where life support systems must endure harsh environments. The study of tardigrades not only illuminates the limits of life, but also offers a glimpse into potential pathways for sustaining life beyond Earth. Bioinformatics plays a crucial role in tardigrade research, allowing scientists to analyze vast amounts of genetic data with precision. By employing advanced algorithms and computational tools, researchers can map the genomes of these resilient organisms, revealing the intricate structures and sequences that contribute to their survival in extreme conditions. This region of study highlights the specific genes responsible for desiccation tolerance and radiation resistance, allowing for a better understanding of their unique adaptations. Data analysis techniques, such as comparative genomics, enable the identification of evolutionary relationships among tardigrades and their relatives. By comparing the genetic material of various species, insights are gained into the evolutionary history and diversification of these organisms. The utilization of machine learning further enhances the ability to predict functional outcomes of specific genetic variations, providing a clearer picture of how they cope with environmental stressors. Phylogenetic analysis offers a visual representation of the evolutionary pathways taken by tardigrades and can shed light on their ancient lineage. This information is essential for unraveling the mysteries of life's resilience. By integrating data from multiple sources, including transcriptomics and proteomics, a holistic understanding emerges, revealing the coordination of biological processes that enable these microscopic travelers to thrive under conditions that would be inhospitable to most forms of life. Ongoing debates surrounding tardigrade taxonomy reflect a complex interplay of evolutionary biology and genetic analysis. Classification efforts have long struggled with the vast diversity within this phylum. Recent advancements in molecular techniques have uncovered new species, challenging traditional taxonomic frameworks. Genetic data reveals significant variations, prompting researchers to reassess existing classifications and consider the implications of cryptic species. These microscopic organisms exhibit remarkable adaptability, leading to discussions about their evolutionary history. Insights into their phylogenetic relationships are reshaping our understanding of how they have survived through extreme environmental shifts. This region of study not only addresses species identification, but also raises questions about the evolutionary pressures that have shaped their resilience. The implications of this research extend beyond taxonomy. Understanding these relationships aids in deciphering ecological roles and evolutionary trajectories. It becomes increasingly evident that the unique adaptations of tardigrades may inform broader biological principles, potentially influencing studies in extremophiles and astrobiology. As discussions continue, the potential for re-evaluating the taxonomy of tardigrades remains open. This ongoing inquiry illuminates the intricate web of life on Earth, showcasing the dynamic nature of biological classification. Through persistent exploration, clearer insights into their lineage and ecological significance may emerge, enriching our comprehension of life's resilience. Research into tardigrades has prompted comparisons to other extremophiles, organisms that thrive in conditions previously deemed inhospitable. This region of study reveals remarkable adaptations across various species. While tardigrades withstand extreme temperatures, radiation, and desiccation, certain bacteria and archaea exhibit similar resilience under high-pressure environments, such as deep-sea trenches. These microbial life forms possess unique biochemical pathways that allow them to endure conditions that would typically disrupt cellular integrity. The study of these organisms highlights a spectrum of survival strategies. Tardigrades employ a process known as cryptobiosis, allowing them to enter a state of suspended animation. In contrast, some extremophilic bacteria utilize biofilms to shield themselves from environmental stresses. This protective mechanism aids in nutrient retention while enhancing survivability during periods of extreme desiccation or exposure to harmful radiation. Comparative studies reveal that these organisms share evolutionary traits that confer resilience, suggesting a common ancestral lineage despite their diverse habitats. Insights into their genetic makeup further illuminate the molecular basis for their survival. This understanding may influence biotechnological applications, where harnessing these mechanisms could lead to innovations in medicine, agriculture, and space exploration. Observations of tardigrades and their counterparts continue to unravel the intricate web of life, showcasing the remarkable adaptability found within the microbial world. Tardigrades play a crucial role in nutrient cycling, contributing significantly to the ecosystems they inhabit. Their feeding habits facilitate the breakdown of organic matter, allowing nutrients to be released and made available for other organisms. These microscopic travelers consume a variety of materials, including algae, mosses, and decaying plant matter. This activity not only aids in the decomposition process, but also enhances soil fertility in terrestrial environments. In aquatic ecosystems, the presence of tardigrades aids in the remineralization of nutrient compounds. By consuming detritus and bacteria, they contribute to the cycling of carbon and nitrogen, essential elements for sustaining life. As tardigrades metabolize organic material, they convert complex compounds into simpler forms, which can then be utilized by other microorganisms and plants. This process ensures a continuous flow of energy through the food web, maintaining the balance of ecosystems. Tardigrades exhibit resilience under extreme conditions, allowing them to thrive in environments where many other organisms cannot. Their ability to enter a cryptobiotic state during adverse conditions, such as desiccation or extreme temperatures, ensures their survival and continued participation in these vital processes. This remarkable adaptability highlights the importance of tardigrades, not just as survivors, but as essential contributors to the health and sustainability of their ecological niches. Educational outreach plays a pivotal role in fostering an understanding of tardigrades and their significance within various ecosystems. Initiatives aimed at increasing awareness often focus on engaging younger audiences through interactive workshops and science fairs, where specimens are showcased under microscopes. This direct observation allows participants to witness the resilience and unique morphology of these organisms firsthand. Curricula designed for schools frequently incorporate tardigrade studies, emphasizing their remarkable qualities as a gateway to broader discussions about extremophiles, biodiversity, and ecological balance. By highlighting their presence in extreme environments such as deep-sea trenches and Arctic ice, educators illustrate the adaptability of life in the face of harsh conditions. Publications and digital media also serve as vital tools for disseminating knowledge about these microscopic travelers. Documentaries and online resources provide detailed insights into their biology and survival mechanisms, reaching audiences far beyond traditional classroom settings. Engaging narratives about their journey through space and ability to withstand radiation contribute to a growing fascination with astrobiology. Collaborations with researchers and institutions enhance outreach efforts, promoting citizen science projects that invite the public to contribute to ongoing studies. These initiatives not only deepen appreciation for tardigrades but also encourage broader discussions about the interconnectedness of life and the preservation of diverse ecosystems. The remarkable resilience of these organisms has captured the interest of bioengineers seeking to replicate their unique adaptations. Their ability to endure extreme conditions, including desiccation, radiation, and the vacuum of space, presents a fascinating avenue for innovation. Techniques inspired by tardigrades may transform material science, enhancing the durability and functionality of products. Among the key adaptations is the production of a protein known as trehalose, which helps protect cellular structures during dehydration. This sugar molecule stabilizes membranes and proteins, allowing cells to survive in a state of cryptobiosis. Understanding the mechanisms behind this protective strategy could lead to advancements in pharmaceuticals and preservation techniques, where stabilization is critical. Incorporating these adaptations into synthetic materials is another promising area. Researchers draw on the molecular architecture of tardigrade proteins to develop coatings that resist environmental stresses. These bioengineered solutions could find applications in various fields, from construction to aerospace, where resilience is paramount. The study of tardigrades continues to unveil insights into their extraordinary survival strategies. As research progresses, the potential for practical applications broadens, enabling the translation of biological resilience into technology. This pursuit emphasizes the importance of nature as a source of inspiration, fostering innovation that harmonizes with the environment while pushing the boundaries of human capability. Key figures in tardigrade research have made significant contributions to the understanding of these microscopic organisms. One prominent scientist, Dr. Thomas Boothby, has focused extensively on the molecular mechanisms that enable tardigrades to withstand extreme conditions. His work has unveiled the protective proteins that allow them to endure temperatures from nearly absolute zero to well above boiling. Such discoveries have implications for biotechnology, potentially aiding in the preservation of biological materials. Dr. Mark H. Stroud has also been a key player in this field. His research delves into the intricate relationship between tardigrades and their environments, exploring how these creatures adapt to desiccation and radiation. By examining their resilience, a deeper understanding of life's potential to thrive beyond Earth emerges, suggesting possibilities for extraterrestrial life. Another notable figure is Dr. Anna the 1000th T. Schmid, whose investigations into tardigrade reproduction and evolution have provided insights into their genetic diversity. This genetic resilience hints at the evolutionary pathways that allow life to persist in extreme conditions. These pioneers collectively shape the narrative of tardigrade research, bridging gaps between fundamental biology and practical applications. Together, their findings not only deepen the scientific community's grasp of these organisms, but also expand the horizons of what life can endure, igniting curiosity about the potential for life in the cosmos. Landmark studies have unveiled remarkable insights into the resilience of tardigrades. Investigations into their cryptobiosis revealed an ability to enter a state of suspended animation, allowing them to survive extreme conditions including desiccation, radiation, and temperatures ranging from near absolute zero to over 370 degrees Celsius. Research conducted by a team using the model organism Hypsibius dujardini demonstrated that these creatures can withstand the vacuum of space. Experiments aboard the European Space Agency's Photon M3 mission showcased their remarkable endurance, as specimens survived exposure to cosmic radiation and the harsh conditions of outer space for 10 days. Genomic analyses have also provided a deeper understanding of the genetic mechanisms that contribute to their survival. Specific genes responsible for protein synthesis during desiccation have been identified, shedding light on how tardigrades stabilize their cellular structures under duress. Additionally, studies have investigated the role of protective proteins known as trehalose, which assist in cellular repair and protection from damage caused by dehydration and other stressors. These findings collectively illuminate the adaptive strategies employed by tardigrades, reinforcing their status as one of nature's most enduring organisms. Their remarkable capabilities continue to inspire ongoing research, with implications for astrobiology and the quest for extraterrestrial life. The fascination surrounding tardigrades has led to numerous myths and misconceptions. One prevalent belief suggests that these organisms can survive indefinitely in a state of suspended animation. This assertion overlooks the reality that, while they can endure extreme desiccation, they require water to reactivate their metabolic processes. Without hydration, survival is limited, as they cannot sustain life in a dormant phase indefinitely. Another misconception involves their purported invincibility. While it is true that tardigrades can withstand extreme conditions such as radiation and extreme temperatures, they are not immune to all forms of harm. Certain environmental stresses, such as prolonged exposure to intense heat or specific chemicals, can indeed be lethal. This highlights a nuanced understanding of their resilience, which is confined to specific ranges of conditions. Tardigrades are often mistakenly associated with being the most indestructible life forms on Earth. This label diminishes the remarkable adaptability of other extremophiles, such as certain bacteria and archaea, which also demonstrate extraordinary survival strategies. The belief that tardigrades can survive in the vacuum of space has also been exaggerated. While they have been shown to endure short durations in such conditions, long-term survival remains uncertain and requires further investigation. Such myths, while intriguing, must be clarified to appreciate the true nature of these microscopic travelers. Public fascination with these resilient organisms has surged in recent years, leading to their prominent presence in science fairs and exhibitions worldwide. Enthusiasts and students alike showcase their findings, often displaying intricate models and engaging presentations that highlight the extraordinary adaptability of tardigrades. Such events serve as a platform for scientific education, allowing participants to delve into the biology and ecology of these creatures. The interest extends beyond academic circles, capturing the imagination of the general public. Exhibitions frequently include live demonstrations where observers can witness tardigrades in action, moving through their aquatic environments. This visibility fosters a greater understanding of the extreme conditions they can withstand, such as intense radiation and extreme temperatures. Educational programs often emphasize their role in the ecosystem, revealing their contribution to nutrient cycling and soil health. These small yet formidable beings are often used to illustrate broader concepts in biology and environmental science. Bridging the gap between complex scientific principles and accessible knowledge, displays frequently incorporate interactive elements, inviting visitors to engage directly with the research process. Artistic interpretations of tardigrades can also be found, reflecting their unique morphology and inspiring curiosity about the microscopic world. Such public interest continues to propel research, ensuring that these remarkable organisms remain a topic of discussion within the scientific community and beyond. Funding for research into tardigrades has seen a steady increase, reflecting their significance in various scientific fields. Grants from governmental agencies and private institutions provide essential financial support for projects aimed at understanding the resilience of these organisms. This funding often allows researchers to explore the genetic and biochemical mechanisms that enable tardigrades to withstand extreme conditions. Collaboration between universities and research centers facilitates the pooling of resources and expertise. Such partnerships enhance the scope of studies and promote interdisciplinary approaches, linking microbiology, astrobiology, and environmental science. This region of inquiry benefits from institutional support, where dedicated programs aim to unravel the mysteries of these microscopic travelers. Funding sources also include international collaborations, which expand the reach of research initiatives. By pooling resources across borders, scientists can conduct experiments in diverse environments, testing tardigrades in various ecological niches. These efforts reveal not only their survival strategies, but also potential applications in biotechnology and medicine. As the body of knowledge grows, the economic implications become apparent. Tardigrades may hold keys to advancements in fields such as materials science, where their unique properties could inspire new technologies. With ongoing financial backing, the research community can continue its exploration of these resilient organisms, contributing to a deeper understanding of life itself. Collaborations in research have emerged as a vital approach in the study of tardigrades. Specialists from various disciplines unite to explore the complex biology of these microscopic travelers. This convergence of knowledge fosters innovative methodologies, enhancing scientific inquiry. Biologists, physicists, and astrobiologists engage in dialogue, bridging gaps between their respective fields. Interdisciplinary approaches allow for the investigation of tardigrades' extraordinary resilience under extreme conditions. Studies often encompass genetic analysis, revealing the mechanisms behind their ability to withstand desiccation and radiation. The application of advanced imaging techniques provides insights into structural adaptations essential for survival in the vacuum of space. In laboratories, researchers are now utilizing artificial environments to simulate extraterrestrial conditions. Such experiments aim to understand how these organisms might survive beyond Earth, potentially informing future missions to other celestial bodies. These collaborations extend to the realms of ecology and environmental science as well, considering the ecological roles that tardigrades play in their native habitats. The synthesis of different scientific perspectives cultivates a richer understanding of life in extreme conditions. This collective effort underscores the significance of interdisciplinary research, illuminating the pathways through which knowledge expands. Through shared resources and expertise, the scientific community continues to unravel the mysteries of these remarkable organisms, advancing our comprehension of life's potential across the universe. Research initiatives are underway to explore the potential applications of tardigrades in various fields. These organisms exhibit resilience against extreme conditions, leading scientists to investigate their genetic makeup. This exploration could unlock insights into enhancing human survival in harsh environments, such as space. By studying the proteins and genes that confer their remarkable survivability, researchers aim to harness these mechanisms for biotechnological advancements. Efforts are also being made to understand the implications of tardigrades' extremophilic traits for astrobiology. Their ability to endure desiccation, radiation, and extreme temperatures raises questions about life's adaptability beyond Earth. Missions targeting celestial bodies like Europa and Enceladus may benefit from understanding how these microscopic travelers withstand conditions previously deemed uninhabitable. Laboratories continue to simulate various extraterrestrial environments to observe tardigrades' responses. This investigation offers a glimpse into how life might exist on other planets, challenging established notions of habitability. Continued research may lead to future missions designed to capture and study these organisms in their native habitats, revealing more about their life cycles and ecological roles. The overarching goal remains to comprehend not only the limits of life on Earth, but also the broader implications for life throughout the cosmos. Each discovery paves the way for a deeper understanding of biological resilience and the potential for life beyond our planet. Research into tardigrades has opened intriguing pathways for human health applications. These microscopic organisms possess a unique ability to withstand extreme conditions, including high levels of radiation and desiccation. Their remarkable resilience is partly due to a protein known as trehalose, which protects their cells during dehydration. This protein's properties have garnered attention from scientists exploring its potential in preserving human cells and tissues. Applications in cryopreservation are particularly noteworthy. Tardigrades can survive frozen states for extended periods, mimicking their survival mechanisms could enhance the storage of organs for transplantation, potentially increasing the success rate of such procedures. This region of study offers promising avenues for medical advancements, particularly in the context of organ scarcity. Research has also delved into the potential of tardigrade proteins to bolster human immunity. Their ability to thrive in environments with harmful levels of radiation may inform new strategies for developing therapies against radiation exposure, particularly for patients undergoing cancer treatments. Further investigation into their genome reveals genes that could be harnessed for improving stress tolerance in human cells. These genes might lead to breakthroughs in regenerative medicine, where enhancing cell resilience could significantly impact recovery from injuries or degenerative diseases. The implications of tardigrades in human health remain vast, reflecting their intriguing biology and adaptability. Understanding the legacy of these microscopic travelers reveals insights into resilience and survival. Their ability to withstand extreme conditions offers significant lessons for various fields, from biology to astrobiology. Within this region of exploration, researchers are studying the mechanisms that allow tardigrades to endure temperatures ranging from near absolute zero to over 300 degrees Fahrenheit. Wolframalids-civil organisms exhibit cryptobiosis, a state that enables them to suspend their metabolism and effectively become dormant. In this state, they can survive environments that would be fatal to most life forms, including exposure to intense radiation and the vacuum of space. This resilience prompts inquiries into the potential applications for human space exploration, where understanding survival strategies could inform the design of future missions. Additionally, the genetic and biochemical pathways discovered in tardigrades hold promise for advancements in medicine, particularly in preserving cells and tissues for transplantation or long-term storage. Investigating their unique proteins, which provide protection against desiccation and radiation, may lead to breakthroughs in biotechnology. This exploration extends beyond mere survival. It touches on the essence of life itself. Insights drawn from these organisms encourage a re-evaluation of the limits of life in extreme environments. The enduring mystery surrounding these remarkable organisms continues to captivate both scientists and enthusiasts alike. Their ability to endure extreme conditions raises questions about the fundamental nature of life itself. Observations indicate that tardigrades can withstand temperatures ranging from nearly absolute zero to over 300 degrees Fahrenheit, reflecting an impressive resilience that few life forms can match. This resilience extends to pressures found in deep ocean trenches and the vacuum of outer space, where they have been observed surviving harsh cosmic radiation. Research into their unique adaptations, such as the ability to enter a cryptobiotic state, reveals complex biochemical processes that allow for such survival. These adaptations involve the production of proteins and sugars that protect cellular structures during desiccation and extreme environmental stress. This region of inquiry not only sheds light on tardigrades but also hints at the potential for life beyond Earth, inspiring astrobiological studies aimed at understanding how life might adapt to extraterrestrial environments. Their presence in diverse habitats, from the highest mountains to the deepest oceans, serves as a testament to life's remarkable versatility. As studies continue, the intrigue only deepens. Each new discovery about these microscopic travelers expands the boundaries of biological understanding, challenging preconceived notions of survival in the universe. The quiet tenacity of tardigrades remains a profound testament to life's enduring spirit.